All right, so in the next section, we're going to take a look at and focus in on one of the main functions of proteins, which is as enzymes. So we can define an enzyme as a biological catalyst, right? Enzymes, they're not always proteins, but very frequently they are. And we talked about catalysts back in the reactions chapter. So remember, a catalyst is something that speeds up a reaction, but is not used up. Which means the catalyst is there at the end to catalyze another reaction. So the thing about enzymes, though, is that they are biological catalysts, and you find them catalyzing nearly all of the reactions that take place in your body. And the way that they work, as we've seen back in, it seems like a long time ago now, is they lower the activation energy for that particular process, right? So if you were to take a look at a normal process without an enzyme, here we have our reactants, and then we have our products over here, right? In order to get from reactant to products, we'd have to overcome this huge hill, or this activation energy, or this barrier to get to the product. But what an enzyme does is it actually lowers that hill or that barrier now, so we don't have to get nearly as much energy to the reactants, because the enzyme actually allows that process to happen with way less energy, and as a result that process will happen a lot quicker than without a particular enzyme. Enzymes themselves have names obviously associated with them, and quite frequently, not always, but very frequently, enzymes in the suffix ase. Right, so for example, you have an enzyme called lactase, which you probably have heard about before. All right, so lactase tells you right away that it's an enzyme, and then the first part of the name tells you what it actually does. So this is an enzyme that breaks down lactose, right? And that's the first part of the name here that tells you what it does. So the ending ase tells you it's an enzyme. The first part tells you what it does, or what it actually works on. So we refer to that substance that it works on as the substrate. All right, so we would call lactose here. So we'd say lactose is the substrate for lactase. In other words, lactose is the thing or the molecule that lactase works on or breaks down or builds up, if that were the case. So enzymes are named typically that way. Sometimes you will see enzymes named based on the type of a reaction that they would catalyze. So for instance, an oxidase enzyme would catalyze an oxidation reaction. And then you'll see some other names for enzymes that maybe don't make a whole lot of sense. They don't have that ase ending. For instance, pepsin and trypsin. These are just older names, and we haven't changed them because they've been known for so long that we just stick with them. All right, but generally your enzymes are going to have that ase ending, and the first part of the name tells you either the type of a reaction that it does, or the substrate that the enzyme is working on. All right, so let's take a look at the mechanism of enzyme action. So how do enzymes work? Okay, so first of all, let's orient ourselves. You have an enzyme, which again is typically a protein. So there's our enzyme, and on the surface of the enzyme, usually a little pocket, a small region, like a pocket or a depression, or just a part of the surface of the enzyme that we call the active site, is where all the action happens, right? And that active site is where the substrate is going to bind to or get attached, right? So we can say then that the substrate binds to the active site of the enzyme. And then, how is the substrate able to stay in that active site, right? Why doesn't it just fall off? Well, it's actually held in place by some of those same interactions we've talked about before. It can be hydrogen bonded to some of the amino acid R groups. It can have some salt bridges to some of the other R groups. It can form some hydrophobic interactions, right? So there are reasons why your substrate actually stays in the active site and doesn't fall off. So the active site, a little pocket where the substrate is actually able to bind to. Once it binds to this active site, so there's your substrate in the active site, then the substrate will undergo, so the substrate then undergoes conversion to the products or a product, depending on what the reaction is, what reaction's happening, and then that product gets released. So once those products leave, then the active site is freed up, which means another substrate can come in and be catalyzed. And that goes on over and over and over again, which is why enzymes are so powerful things, right? They're able to catalyze many reactions over and over and over again. Substrate comes in, gets converted, the products leave, another substrate can come in, and that can repeat itself multiple hundreds, millions, even billions of times a second, depending on the enzyme that you're working with. All right, so we can write a little mechanism for this. Your enzyme and your substrate combine together to form an enzyme substrate complex, which we abbreviate as the ES complex, and you can write that here. This is usually a reversible process. So E is enzyme in our little diagram. So the enzyme here is E, the substrate is S. All right, so your enzyme and substrate come together. We form our enzyme substrate complex. The enzyme does its thing, converts the substrate to product, and then the product here, P, leaves. And then that free enzyme now can go back in and start this process over and over and over again. Right, that's a simple way by which we can describe an enzyme catalyzed reaction. So if you take a look at a specific example, we choose the molecule that we talked about before. So when the disaccharide, the sugar lactose, is hydrolyzed, right, the enzyme, which would be lactase, right, the lactose can come in and they can form an enzyme substrate complex. So a lactase lactose complex, the enzyme does its thing on the lactose, and if you remember, lactose is made up of glucose and galactose, and those two things get released. So we end up then with our free enzyme lactase, and then our products glucose and galactose. All right, these are our products. So the products leave. There's our free enzyme. Active site is open. It can come back around and it can repeat that process over and over and over again. Right, as long as there's lactose present. For example, if you just drank a bunch of milk. This is how this enzyme catalyzed hydrolysis of lactose and pretty much any other process would happen. So there are a couple models that describe in a little bit more detail how the enzyme is able to do that. The first model is known as a lock and key model. And it may be sort of self-explanatory, but sometimes it's a little bit difficult to make some sense of it. So in this lock and key model, we can think of the enzyme as the lock, and then the key is going to be the substrate. Right, so for instance, if you have and we'll just do it in block diagrams just because it's difficult to draw proper three-dimensional shapes. So if I have my enzyme that looks like this, and then here's my active site. Okay, this is my enzyme, and that's the active site. Right, you'll see that the active site has a particular shape. Well, my substrate then, in order to fit into that lock, would have to have a shape that resembles that. Right, so in this case, it's triangular. So this is my substrate, and this is going to have a shape. Substrate has a shape that is, we say that it is complementary to the active site. Right, so this then is able to come in and fit right here into that little pocket. So there's only that shape, that key, that can fit into that lock. That's the idea behind the lock and key model. Obviously, if you have something that has a very different shape than this, right, that is not going to be able to fit into that lock. So that's the lock and key model, which is predicated upon the both the enzyme active site has a very rigid shape. Okay, so the active site is rigid, kind of like a lock, and there's only one shape of the substrate that can actually fit in there. So that type of model can be used to explain the behavior of some enzymes, but not all of them. There's a slightly different model known as the Induced Fit Model, which instead says that the structure of the enzyme, specifically the active site, is not rigid, but it's actually flexible. And it actually can change its shape, so it adjusts to the shape of the substrate in order for it to bind properly to the substrate, right? So what that suggests then is that this enzyme can actually bind to a range of substrates in terms of their shape. As the shape changes, it can actually improve the catalysis or the breakdown of the substrate. All right, so if you take a look here, this is maybe a better diagram of this process. So here's your, well, this is the lactase lactose example. So there's our enzyme lactase over here, and then you can see the active site has a shape that looks like, mirrors a triangle, but the shape of the substrate, right, the lactose itself, is not exactly complementary to the shape of the active site. But when the substrate comes close to it, right, what you'll see here is that the shape of the enzyme, so the enzyme active site, is flexible. Right, you'll see it's not triangular as it was before. It actually is able to adjust so that the substrate binds to it, forms the enzyme substrate complex. The enzyme does its thing, breaks this bond right here between the glucose and the galactose, releases those products, and then the enzyme is freed up, and it can repeat that process over and over and over again. All right, so this would be a good example of the induced fit model, right, where the enzyme's active site is flexible and it adjusts to the shape of the substrate. So when we take a look at the next section, we're going to see how the activity of enzymes can be affected by several different factors.